So as Josh said, I'm Lindsay. I'm a co-founder and CTO at a startup company called Talus Bio. And as he said, we do mass spectrometry proteomics, but we also do a lot of other omics as well. And one of the things that I'm thinking about a lot these days, as it sounds like a lot of you out there are also working in the space of kind of, large sample sets, juggling a lot of different data from a lot of different sources, a lot of different instruments. When we go into some of these studies, I want to tell you about how we're thinking about prospective system suitability and quality control, as opposed to, collecting all of your data and then going back and trying to curate it, doing these things as the samples are being acquired and processed. So at Talus Bio, we do proteomics. Specifically, we look at nuclear proteins. We call it the regulome. So that's just any one of the proteins that is interacting with DNA, with the genome, and in some way controlling gene expressions. This can be anything from, the canonical transcription factors, all of the cofactors that bind to transcription factors, even, the transcriptional machinery like RNA polymerases, or structural proteins that are binding and packaging DNA like histones. All of these things are kind of what we're interested in from a drug discovery standpoint, specifically for pediatric cancer. And kind of our idea, our thesis is that transcription factors are these master regulators that can either control the fate of a cell, for example, a stem cell, through a program of transcription factors is differentiated into a T cell. Different transcription factors then differentiate that T cell into an inflammatory T cell. However, there's another program of transcription factors that could turn that T cell into leukemia, into cancer. So our thesis at Talus is, well, if we know all of the transcription factors that can influence a cell to become cancer or to become properly differentiated, then we can manipulate those proteins through small molecules to either cure cancer or prevent it from happening in the first place. So when we're measuring data, we're measuring the proteins, and we're looking for the difference between the proteins in a cancer cell, for example. We'll have some subset of transcription factors that are binding to DNA. We measure that. And then we treat those cells with a particular drug candidate, a particular small molecule from, one of these giant small molecule libraries with thousands, tens of thousands of small molecules that need to be tracked in sample managers, like presented before me. We treat those cells and we see what transcription factors have been affected or influenced by those small molecule probes. Compare these two samples, and now you have potential small molecules to follow up with drug development efforts. You also get bonus data in this that I'm not going to really talk about in this, where, not only the transcription factors that were inhibited by the drug, but you also kind of get a picture of which drugs might have off-target toxicity or off-target beneficial effects. So our data ends up kind of looking like this for every single one of our samples. Here I'm showing a example data set that was a time course. We look at proteins that are just floating around in the nucleus, not bound to anything. We're looking at proteins that are bound to open chromatin, where DNA is being transcribed and expressed, and looking at proteins bound to heterochromatin, where the DNA is closed and genes are not being transcribed anymore. So we end up with this kind of giant matrix of data where we have on one axis different samples, different time points in this experiment, different time points of the same drug. But you can imagine this being, instead of time points, being 6 different drugs and a control. And then along the other axis, we see all of the other proteins that we've measured. And each individual point, a combination of a protein and a sample, is a expression value, an abundance of a particular protein. So this would be maybe one data set, one of these small experiments, but what our goal is to build a giant data repository of all of these. So testing, tens of thousands, millions of compounds of small molecules, look at hundreds of different cell lines, different cancers and healthy tissues, and then measuring all of the different gene regulatory proteins through these targeted mass spectrometry methods that are supported by Panorama. So 1,000 of these gene regulatory proteins. So we have this giant repository of proteomes. We at Talus use those to make predictions. After those predictions are made, we go out and actually test those compounded conditions, use the results of those tests to refine that model, and the database keeps getting bigger. The database keeps benefiting from this cycle, and we use this to develop drugs for these cancer targets. But as many of you have probably experienced, doing this is great and a lofty goal, but it really depends on the quality of that original data set. Garbage in, garbage out. So that data set really needs to be well curated. And we could do that retrospectively. We could collect thousands of samples of data and then go back and try to batch normalize and throw out outliers or repeat batches that looked like they didn't run correctly for some reason. But why don't we just do this at the beginning to start? Instead of going back to do this, why don't we catch those before they even make it into the database? And this is a problem, at least in mass spectrometry and proteomics, that is not new. There's been a lot of efforts to analyze lots of samples that did not start with these prospective study designs and instead tried to retrospectively come up with interesting biological conclusions. So this is an example of a data set where they did proteomic profiles of 2 cancer types. And if you're familiar with reading dendrograms like this, you can just kind of draw a horizontal line, and where that horizontal line cuts these branches going down, that's how many clusters there are. So if we draw a horizontal line at the top here, there's 2 clusters. Well, they profiled 2 cancer types, so that should check out, right? But it turns out they prepped each one of their cancer types with a different protocol, because one of the types of cancer subtype didn't process, the samples didn't process correctly with the first protocol. So they said, oh, cool, we'll like make a protocol to process these samples. And they didn't stop to think, well, those changes in protocol can obviously affect the data that's coming out. So they have perfectly confounded data because they use 2 different protocols on their 2 different sample types. Another example, proteomic profiles of 5 different cancer types. And if you draw a line across here, you would expect to see 5 groups for the 5 cancer types, but now their clustering suggests 6 different cancer types. And this is not a problem with their protocol this time, but if you went back and looked in their metadata, the time stamp on their data acquisition, these 6 clusters perfectly confound with the run order. So they loaded up samples on their instrument, ran their instrument. Maybe they had to stop in between some of these samples to clean the instrument, or a different technician or a different batch of reagents. Whatever it happened to be, they ended up having perfectly confounded run order with their original sample design. So mass spectrometry, just like any other measurement system out there, is not inherently quantitative. You have differences from sample to sample, and you're hoping that the differences you're measuring from sample to sample are due to the underlying biology. But it could be the operator. If you have a large enough group, you have multiple technicians, multiple operators that are manipulating samples. You might be fortunate enough to have multiple different instruments, and different instruments such as mass spectrometers, but could be anything else, can also introduce variability across your different sample types. So capturing information, we just heard about capturing metadata at the sample level, but also capturing information at the instrument level is incredibly important. So not only the operators preparing the samples, but also the operators running the instrument. So how do we ensure data quality? Long story short, too long, don't stay for the rest of the presentation. Monitor process controls at each critical point, assess your system suitability regularly, and capture as much metadata for each sample as you can. So mass spectrometry proteomics is a very involved workflow. Every biological sample prep involves a lot of hands and a lot of steps. Not to go into details here, but just to say that in the workflow for mass spectrometry proteomics, we're doing a lot of manipulation at the front to extract protein. We enzymatically digest it. There's a couple other steps. We end up with peptides ready for mass spectrometry. But then on the instrument side, we then separate peptides using liquid chromatography. And then inside the mass spectrometer, there's several steps that are specific to the system and at the time you're operating before you get the data acquired at the end. So these are kind of two different things to be looking for. The top, when we're actually physically manipulating the sample, that's quality control, that's the sample variability itself. And then on the bottom, when we actually get to the instrument system, the analytical system, the LCMS, that's system suitability. So that's the instrument variability. So the top, the physical process of manipulating the sample, and on the bottom, the instrument that you're using. Typically, workflows should be, if you're not, they should be using these system suitabilities. No matter what you're using, whatever analytical system, you should have some sort of test sample that is the same always. You have this test sample in the freezer. It's, it's some E. coli genomic DNA, or it's in proteomics. A lot of times people use like a HeLa cell lysate proteome. Whatever it is, you have like big batches of it and you throw it in your freezer and you run it interspersed with your samples so that you can check your system is giving you the right answer that it should be giving you. And just to plug one of my absolute favorite papers of all time that did this with mass spectrometry, this is St. John Williams et al. 2017. They did a metabolomics and lipidomics study where they set up their 96-well plate layouts to include almost a third of the plate, a fourth of the plate, just using these samples to assess not only the sample quality with QCs, but also doing calibration curves and including reference samples so that as they're going batch to batch, they can run these initial samples and determine quickly before they run 96-well plate worth of samples. Within the first 12, 13 samples, they can determine, is this plate a go or a no-go? Do I waste the instrument time and all of the reagents processing this plate, or is there something present in those QCs or in those system suitabilities that tells me I have to hit the stop button and figure out what's going wrong before I run this plate? So the way people in mass spectrometry used to do this was very painful and manual. And every time you would acquire data, you would put it into this software called Skyline, which is a great software. And it's super awesome for analyzing targeted mass spectrometry proteomics. But you can imagine if you're doing a very large study and you have hundreds of samples that you want to be checking, manually, every time you run one of these, you have to log into the instrument and go file, import results, and import your results file. It's clearly not conducive to a low barrier of entry, especially if you're an overworked technician or a grad student. You're probably not going to do this as often and as frequently as you should be doing it in order to be monitoring effectively. So what the LabKey team did, they have under the LabKey umbrella, there's this panoramaweb.org. So LabKey Panorama takes those mass spectrometry data analysis documents and puts them in the cloud, puts them on this website. So now, instead of everybody kind of pouring in their time and effort into mass spectrometry assay development and doing these manual quality control documents, now there was this relational database to store these documents and aggregate and organize the documents. And then they went a step further from that, because now it's like, okay, but this doesn't solve my problem of file, import my results every single time, and then I can share it on the website. That's great. But they went a step beyond, and the team made this small piece of software called AutoQC that sits on your mass spectrometry instrument and just kind of runs in the background without you doing anything once you've set it up. And now all of that monitoring is done for you. All that data analysis is done for you, and it gets pushed up to Panorama. To show you a little bit about how we're using this, this is like exactly what it actually looks like on the Talus website. So I have 3 different instances of this setup. Not all of them are running right now, and I can tell because AutoQC on the Panorama Web website will have this red dot next to it or a green check mark next to it. So I, as CTO and kind of PI of this lab, I can quickly log in and then see, why aren't we running digestion efficiency and nuclear enrichment quality controls? Or I could log into system suitability and say, all right, everything looks like it was running great as of yesterday, at least. Each one of these individual AutoQC kind of instances, you can click on these. So if I click, for example, on system suitability, that'll take me in to that specific QC, this specific system suitability, and I can check on different quality control or system suitability metrics. So for example, with mass spectrometry, the instrument measures mass to charge. So one of the things that you are inherently interested in with the performance of your system is what's the mass accuracy. If the mass accuracy is starting to go crazy, it's probably time to calibrate the instrument. Other labs, when you're doing this, they might just automatically calibrate every other week or maybe even every week and not really care if it's actually necessary or not. But over calibrating your system can also cause problems. So only calibrating when you need to. I can just log into Panorama Web, do a quick check, be like, oh, it's starting to creep up past the parts per million that I'm comfortable with. In these kind of graphs, I had mentioned and kind of glossed over, I set up 3 different instances of AutoQC. Each one of those instances is monitoring different analytes. So the system suitability is monitoring a specific set of analytes, and nuclear enrichment and digestion efficiency are monitoring different sets of analytes. So you can kind of customize the way AutoQC is being used, either with the analytes that are present in the analysis document or by the types of files that it's monitoring for, because you can set up where exactly data files are being acquired for AutoQC to watch and automatically upload. So you can kind of customize this if you're a little bit thoughtful ahead of time with that prospective study design. Okay, what are proteins that I expect to always see? What are analytes I always expect to see in my samples? And do I expect them to change or not? So if you set up your document with particular contaminants or something, now you can watch whether those contaminants are starting to creep too high. If they're starting to increase, maybe that's another quality control issue that you need to shut everything down and say, why are these contaminants starting to increase in my samples? One of the other features that's kind of nice on the tabs at the top of Panorama Web, I was just kind of showing you the dashboard first, but there's also runs. Annotations is something that we like using to keep track of the history of our instrument. So you might see a whole bunch of things noted in our annotations, especially if you're a mass spectrometrist. Some of these things look painfully familiar, like changing the column, changing the ion transfer tube. But now as kind of a manager, I could set this up for all of my instruments and very quickly look down and say, okay, well, we haven't cleaned this instrument since, August. That's actually not too bad. That's pretty good. But if we hadn't cleaned it since, August of last year, maybe that's something that I would pull up and say, okay, we should probably get that scheduled. Speaking of things that are now kind of no-brainers and just kind of automatically done for you, I can log into the website and check all of these things, but I don't even have to. You may have noticed the little email icon next to these. You can set up notifications to just be sent to your email. So Panorama will automatically email you if the most recent samples that are being analyzed have any outliers. So you can set this super stringent and say, notify me immediately if there's even just one outlier, because these are very precious samples or something. Or you can set this to be a little bit more liberal and say, maybe the last 3 samples started looking not so great, then notify me. So we're just about out of time. I want to save a little bit of time for questions. Just to recap, how do we ensure data quality, monitor process controls, assess system suitability, capture metadata? So this is the team that did the majority of this work when we got set up. but I'd be happy to take any questions about how we're using AutoQC for these types of things.
Yeah, please go ahead and submit questions through Zoom. And kind of starting out, actually on almost one of your last slides, Lindsay, a question about how you use the AutoQC reporting in terms of the number of outliers, kind of the summary that you mentioned on that email threshold. If you guys kind of talk about that.
Yeah, so this is also, I didn't even talk about this, but this gets into the idea of guide sets in AutoQC and in Panorama. So when you're using PanoramaQC, you should run, like some like 7 or 10 samples that you think look good within your acceptable realm. Like if you're going through manually curating your data, you would not be concerned about that level of variability. You set that in Panorama as your guide set, and then outliers are determined from there. So in that guide set, if you're like anything outside of this realm of variability is unacceptable, then I would set it to say 1 outlier. However, if you're like, yeah, I could handle a little bit more fuzziness in this, you can set those outliers to be a little bit higher. So guide sets on there. There's a really good tutorial on the website on Panorama. I'll share my screen really quick just so that you can see where this is. But yeah, if you go to the website, to the Panorama website and look in the documentation, there's really great documentation on the side there. Vigisha's done a great job of that.
And then I know from talking with other groups that, especially first-time adopters often kind of struggle with being overwhelmed by outliers. If you could maybe talk a little bit about how you kind of refine or, either the set of analytes that you're monitoring or, excluding problematic samples so they're not messing up your expected range, things like that.
Yeah, that's definitely a huge thing. And I'd say definitely at first, maybe just kind of play around with it a little and see where your realm of acceptability is. Because if you launch straight in and you want to set up a guide set, and you're running, 10 samples back to back to back to back, that's probably artificially going to look better than your real data collection is going to look. I would say first, collect some longitudinal data or have some repository of data to pull those 10 samples from over the course of some months to see where your actual variability is. This is another thing I didn't show on Panorama Web, but there's another tab at the top there for Pareto plots. And those are really great to show, where is the largest source of your variation coming from? Is it coming from your liquid chromatography system, or is it coming from the fragmentation? But again, this is on the website too, and I can share some links if you want to reach out, either Twitter or email. I can get those to you. I'd say do that. And then also when you're choosing those analytes, do the same thing. Whatever set of analytes you think you want to use, look over some historical data, at least a couple months worth across the different sample batches, and make sure that they're stable, present in all of your samples, and you feel are reliable, or they're representative of what you want to measure. If it's a contaminant that you only see every 3 months, maybe that's not a great analyte to use as a system suitability.